Welcome back. In this portion, we are going to be learning about the plasma membrane or the plasmalemma of the cell. And so the plasmalemma is not like a Ziploc bag inside which we have all the various organelles and the liquid that keeps everything in there and keeps everything outside. But the plasma membrane actually controls what goes in and out of the cell. It's very fluid. It's not passive. Okay. This is called a fluid mosaic model. Not important you learn that name, but just in case you're reading it in your book or someplace else, that's the model that we're going with. What the plasma membrane does is creates a different environment inside the cell than outside the cell. Okay, so think of it kind of like being like the structure of your house. Okay, what the environment inside is not the same as outside. In the winter, it's a lot warmer inside the house, and yet you still have windows and you have doors where stuff can come in and out. Some things require doors, some can go through windows, some things can go through the cracks around there, because like ants can come in. And so think of the plasma membrane sort of like that, creating separate environment. And when you look at this picture, it looks kind of complicated, doesn't it? We're going to break it down so that you can get it. So the first concept you need to understand is something called the phospholipid bilayer. So let's just look at those words. Okay, everybody recognize the word lipids. Okay, what are the building blocks of all lipids? That's right, fatty acids. And then we have our prefix phospho, which refers to there's going to be a phosphate group, which is an electrolyte. It's got a negative charge to it. Okay. Then looking at bilayers, well, what does that prefix bi mean? What does it mean in bicycle? In bicycle, it means that there's two wheels. So bilayer means there are two layers going on. So here is a phospholipid. We have two long fatty acid chains, and then we have a phosphate group. Because it has a negative charge, we are going to have another molecule attached to it, which has a charge to negate the charge. And so what this does, since we're not in chemistry, we're not going to be drawing it like this. Basically, the phosphate and other group, that's considered the head of the phospholipid. So we just draw it as a ball. And then the fatty acids are the tails. Okay. Now, think for a second. When you're washing dishes, lipids, fats, and oils, do they like water? They don't, right? You have to use the detergent, the dish soap in order to wash them off. Okay. These fatty acids, they don't like water either. But yet your body is 2/3 water. So what do we do about that? Well, if you were to take a handful, pop all these phospholipids in your hand, and you were to dump them into water, what happens is they spontaneously form a phospholipid bilayer, okay? Where the heads, which like water, are in the water, and the tails, which are the lipid part that hate water, are protected from the water. And so we have one layer of phospholipid molecule and a second layer. So this is our phospholipid bilayer, where we have a watery environment on one side and we have a watery environment on the other side. And where the fatty acid tails are, there is no watery environment. So the head part, we use the word hydrophilic. Hydro meaning water, philic means it loves water. Okay, so we have hydrophilic heads on the watery side. So on the side facing inside the cell, because inside the cell we have that cytosol, which is mostly water, and we also have it on the outside of the cell, which because our environment is mostly water out there as well. And then our tails, the parts that hate water, we call them hydrophobic. Remember, phobia, fear of, arachnophobia, you don't like, you hate spiders. Well, hydrophobic tails, those fatty acids don't like water, so they're on the inside. So this is what our phospholipid bilayer looks like. And so here's an example of a photomicrograph from a, and what that means is a photograph taken from a microscope. So stop and think for a second, what type of microscope would this be? Would this be a light microscope, a scanning electron microscope, or a transmission electron microscope picture? Yeah, this is a transmission electron microscope picture going on. And so we have a blue cell here and a green cell there, just randomly colored so you can tell the two cells, okay? And this right here, okay, sorry, I said that wrong. We have, oh, I'll just leave it there. Right here, these are all the heads. And these are all the heads over there. So we have, and right here, the brown part, that's between. So we have this phospholipid bilayer going on. Now, in addition to the phospholipids, we have more lipids in the membrane, specifically cholesterol. And so cholesterol basically has this structure, which you'll learn when you take chemistry. You don't have to know it for this class, but all of these yellow things is this cholesterol. And the amount of cholesterol you have interspersed among the phospholipids determines how rigid it is. So more cholesterol, more rigidity, less, more fluid, flexible kind of stuff. And lipids on the outside can pass through this lipid portion of this membrane very easily. Now, in addition to lipids, does everybody recognize the phospholipid bilayer? So in addition to the lipids, which we're not showing on this portion, we have proteins, okay? And proteins can be divided into two big categories. The first are those that go all the way across the phospholipid bilayer. So they're called transmembrane, which means across the membrane. They're also called integral. In other words, because they are stuck in the phospholipid bilayer and they cannot be removed. And so there are a lot of different types of these. So anytime this semester or in physiology where you see or we talk about channels or carriers or gates or receptors or messengers, those are all transmembrane proteins that are going to stay. They're not going to be removed. So that kind of alludes to the other type of protein, which is called a peripheral protein, which can be on the outside of the cell, attached to the heads on the outside of the cell, or attached on the inside of the cell. And they have a loose attachment, so you can kind of like, you know, flick them off. And this is what enzymes and some of our cell junctions we're going to be talking about later on. So what do all these structures do then? They allow the cell to have what's called selective permeability. In other words, it can be a little bit of a snob. It can decide what comes in and what doesn't come in. So some things, they can come in. So some very small molecules that are hydrophobic. What does hydrophobic mean? It means they don't like water. Very small hydrophobic molecules can cross easily. So this is stuff like oxygen, which is great. Also nitrogen molecules, carbon dioxide as well. In addition, some uncharged polar molecules like water, ethanol, that's your booze that you're drinking, they pass easily as well. However, larger molecules, certain ions, certain things that aren't soluble in lipids, they can't cross unless there's a transmembrane protein, unless there's a carrier or a channel or something through which they can pass through the phospholipid bilayer. So we can, your cell can control which of those are entering or exiting. Remember, it's a two-way street. Whoops, two-way street. So when we're talking about movement of things across the membranes, there's a whole vocabulary, half a dozen different words we're going to use. So the first term we're going to use is passive transport. Passive transport means there's not going to be any energy required for this to move. And there's a couple of different types of passive transfer. The first is called diffusion. And so in all types of passive transfer, basically something is going to move from where there is a higher concentration of it across the membrane to where there is a lower concentration of it. Okay. So this is what you see when you put your tea bag in the water. You can see the tea moving away from the tea bag. That is just passive transfer, specifically the type known as diffusion. Now, when we are talking about the passive transport of water, which is commonplace considering that our bodies are 2/3 water, we have a one-word term for that, and that is osmosis. Okay, so osmosis is not sleeping on your anatomy textbook and waking up in the morning knowing it all. Osmosis just means the diffusion of water. In other words, the movement of it from an area of higher concentration to lower concentration. In addition to diffusion as an example of passive transport, the other type is what is called facilitated diffusion. We are still going from high concentration to low concentration, so going down a concentration gradient. But in this case, it's one of those molecules or substances that requires a carrier, a channel, or a gate to get across. So it requires one of those transmembrane or integral proteins, but no energy is used. So in contrast to passive transport, we have active transport, which does require energy. Will also require transmembrane protein. Okay, so there's our active diffusion, and our energy source is ATP. So we're going to be talking about this later this semester when we're talking about certain pumps to move things, because they're all going to be requiring ATP. So that's great for our smaller molecules, but if we have something big that needs to move, we need a different process. Like for instance, you've eaten your piece of steak and now you've got some big stuff. How are we going to get that stuff inside there? Okay. Or if you're a one-cell organism in that pond while you're a paramecium and, oh, look, here's breakfast. How are you going to get breakfast inside you? Okay. Well, we have two different processes. So stop for a second. Freeze your thing and try to figure out what does endocytosis mean versus exocytosis. Look at the prefixes, endo and exo, and look at the roots, cyto. Okay, that's right. What does cyto mean? Cell. What is exo? Have you ever seen exo as in an exit sign? That's right. It's where you're leaving. So exocytosis is how stuff would leave a cell. And so the contrast of that would be endocytosis, which is how stuff enters the cell. So for this, I want you to stop and think for a second. Did you have breakfast today? Or did you have dinner last night? Okay. What are the verbs you would use? So what did you have? You're going, well, I had a burger. Okay. So what verb do you use for consuming that burger? You use the word eat, right? Okay. And what happens if you're thirsty? Do you eat a beverage? No, you drink a beverage. Okay. So both of those are examples of putting something in your body. Okay, it's not endocytosis, but I want you to realize in your body, you talk about eating and drinking for putting stuff in. So you got two verbs and you're not complaining about it. So with endocytosis, we're going to have two different types, one for the solid stuff and one for the liquid stuff. So I don't want to hear you complaining about that either. So for the solid stuff, the specific type of endocytosis is known as phagocytosis because phage means to eat. Okay. So when there is a solid particle cruising by your cell, the cell plasma membrane will surround that and engulf it in a process known as phagocytosis. On the other hand, if there is liquid, and there can be stuff dissolved in the liquid, it will not do phagocytosis, but it does the same process, but it's called pinocytosis. So think like wino, only change it to a P. Pinocytosis. Yeah. So that's how things are entering the cell. And then the opposite of that, here we have our little structure, and it has things that we want to get rid of. And notice this has a phospholipid bilayer, just like these had phospholipid bilayer from the plasma membrane. And so the phospholipid bilayer comes along, fuses with the plasma membrane, and then it's kind of like popping the zit, getting rid of the material the cell no longer wants through the process known as exocytosis. And I want to point out that this is a lot more complicated than that simple diffusion or facilitated diffusion. So this requires ATP. So when we go back to where we started over here, this was the simplest, going down a concentration gradient. Then we required some protein carriers, but we still went down a concentration gradient. Then we went up a concentration gradient and required ATP for active transport. Over here, we're still requiring ATP. We don't need carrier molecules anymore. And it doesn't matter if we're going from high to low or low to high. We are doing endocytosis, specifically phagocytosis and pinocytosis, or we're doing exocytosis. All three of which require ATP. So at this point, I want you to take a piece of paper, freeze it, draw a typical plasmalemma, and then turn it back on when you're done. Give you 1 minute for drawing. Okay, so how'd you do on that? All right. So yes, I normally do this in class, and as I'm walking around looking at what people did, I noticed that like 20% of the class will have drawn something like this. All right, this is not a plasma membrane. And some of you who drew this then realize, oh yes, I made a mistake. So then you start trying to draw plasma membrane where you can't see anything because you drew it wrong. In order to get credit, the first thing you have to do is read the question and answer the question being asked. If this was a test or a quiz question, I say draw a typical plasma membrane, and you draw this or this, you don't get any credit. What you need to draw is some type of stick figure of something like this, okay? So this course, super important to pay attention to the details in order to be successful. With that, we're done with the plasma membrane, and that's the end of this lecture. So I will see you again shortly as we continue on learning about the cell. Thank you for all your hard work, and I'll see you soon.